I guess I'm good to go. Good morning. I'm used to presenting to mostly people online, but to introduce myself, I'm Tobey MacDonald. I'm the Director of Pediatric Neuro-Oncology at Children's Healthcare of Atlanta and Emory University. And today I'm going to tell you a story that really spans my entire professional career. So it's really a personal story as well for me. It is about circulating tumor cell clusters as potential novel biomarkers for minimal residual disease and treatment resistance in medulloblastoma. So medulloblastoma is an embryonal tumor of the cerebellum. It's the most prevalent malignant brain tumor in childhood. Virtually all deaths are due to metastasis within the central nervous system, and craniospinal radiotherapy is the standard treatment we use for preventing and treating metastasis in all non-infant patients. However, long-term neurocognitive deficits occur in a majority of patients as a result of this treatment. And survival remains poor for those with metastasis at diagnosis, which is defined by either positive MRI or positive CSF cytology. So back 25 years now in my early career, that was a problem that I was hoping to address. And my hypothesis was that metastasis is a molecularly driven event mediated by the tumor cell. Now, at that time, it was the advent of comprehensive gene expression profiling, which pales in comparison to what we can do today. But the Affymetrix human gene FL microfluidic chips were available for comprehensive gene expression profiling. I say comprehensive, about 1,500 genes were on this chip. And at the time, we knew that medulloblastomas were small round blue cell tumors that look very similar to one another, and that MYC amplification appeared to be associated with greater rates of metastasis. But that was about it. And when we did this gene expression profiling experiment, you can see on the right of the diagram in red, the lower right, increased expression of genes within the metastatic medulloblastoma population. And although there were quite a number of genes there, the most prevalent interacting genes within a common pathway were all within the platelet-derived growth factor receptor RAS/MAP kinase pathway, which we knew at the time also induced MYC. So I put my hands together and thought, great, we've got the answer started. This was parlayed into a KO8 application and then further into my first R01. And multiple people, including us, contributed a number of studies supporting PDGF receptor and RAS role in metastasis of medulloblastoma. But clearly, it's not the whole story. There are multiple interacting signaling pathways. There are some who believe the cancer stem cells are critical to this process, whether they are the cells themselves that are mediating metastasis or influencing. And then, of course, how that is influenced further by interactions with the tumor microenvironment. And I'll just put aside, I put STAT3 in the center here because that is now one of the more focused areas of my research because it does play an important role in all three of those areas, but that's for another day, another talk. To further complicate or make complex this story, a consensus of the molecular landscape of medulloblastoma was derived in 2012. And based on genetic alterations and gene expression differences, medulloblastoma could be categorized into four major molecular subgroups, those that are Wnt-driven, sonic hedgehog-driven, and then group 3 and group 4, which don't have a particular oncogene, but also are now considered to be comprised of the same disease along a spectrum of differentiation within the cerebellum. But what's important about this slide is that both between the major molecular subgroups and now subdivided within those subgroups, between and within, metastasis rates vary wildly. Wnt has the lowest rates of metastasis, ranging 9 to 21%, and group 3 has the highest, ranging from 20 to 43%. So clearly, no one pathway or molecular alteration is driving metastasis in this disease. The capacity is present throughout the molecular subgroups, and likely there is an influence based on your genetic background that facilitates the process. Well, with that, we know that subgroup now is one of the most powerful predictors of outcome, but metastasis still remains the most powerful predictor of outcome among all the subgroups. And so now when assigning risk for treatment of medulloblastoma, it's essentially down to molecular subgroup plus your metastasis status. Those with metastasis or particular genetic alteration will be bumped up to high-risk disease, and that equates to an increased craniospinal dose of 3,600. This results in increased impairment in neurocognitive function subsequently. So following radiation, the standard treatment would be adjuvant chemotherapy, but despite this very aggressive treatment strategy, survival also varies widely between the molecular subgroups, with Wnt having the highest survival, greater than 90%, and group 3 having the lowest, with 40 to 65%, which makes sense since the association of metastasis with the group 3 tumors. So what did change, however, was in 2018, a challenge to the dogma of how medulloblastoma metastasizes. It had been thought that the tumor cells break away from the primary tumor in the cerebellum and then either actively or passively float through the cerebral spinal fluid and then eventually seed along the leptomeninges. So Michael Taylor's group, and this is elegant preclinical study that can only be thought of, I think, by a surgeon, where you took one mouse on the left and injected medulloblastoma cells into the cerebellum, waited a couple days, and then anastomosed a second sort of recipient mouse through the circulation and let them sit together, join for about 4 to 6 weeks, separate the donor mouse, if you will, and look at the mouse that was anastomosed to the mouse that had the tumor cells injected into the brain. And he was able to demonstrate that leptomeningeal dissemination was present in the recipient mouse, therefore confirming, at least in the preclinical status, that medulloblastoma metastasis route could be hematogenious. Further supported this in humans by a flow cytometry study of blood from 6 medulloblastoma patients, and it detected NCAM, or neural cell adhesion molecule positive, CD56 positive cells, in CD45 negative cells. So these were negative for hematopoietic cell lineage, but positive for NCAM in 3 of the 6 patients. And these were believed to be the putative circulating tumor cells. Now, I challenge that to some degree because Michael didn't further molecularly characterize these cells. And it is possible that dendritic cells and immature NK cells may also be CD45 negative and CD56 positive. At that time, we were also thinking of the same idea of circulating tumor cells in mice, at least on the preclinical level. And rather than injecting tumor cells, we use a genetically engineered mouse model to avoid the artifacts that could occur by injecting tumor cells and causing artificial dissemination. So we use the SMO-A1 mouse. This is a mouse in which the precursor cells of the cerebellum have oncogenic smoothing constitutively activated, resulting in sonic hedgehog medulloblastoma formation. And we crossed that mouse with a GFP reporter mouse, so only the tumor cells were labeled with GFP. And you can see on the right, looking under a dissecting microscope, that we can easily visualize metastases along the spinal cord. And then we looked whether by flow cytometry in this case, we could sort out GFP positive cells in the blood as opposed to the normal cells and count the background and see whether GFP positive cells resulted in increased metastasis that we could visualize, at least through the dissecting microscope. And that is indeed the case, where the higher the levels of GFP positive cells in the blood, again, supporting the notion that Michael put forward that hematogenic spread is possible. These mice with the higher levels, and we had an arbitrary cutoff of about 5 GFP positive cells per 10,000 cells in the blood, seemed to have a significant increase in leptomeningeal metastasis. Okay, so 2 mouse models suggesting this occurs. How about in humans? Unfortunately, at that time, there was no assay or biomarker to detect minimal residual leptomeningeal disease. And the majority of MRIs and CSF cytology examinations in our patients are negative throughout their treatment, despite leptomeningeal relapses occurring up to 10 years after completion of therapy. So this leads to the need for liquid biopsy. And in this concept, because these cells are so rare, we need to be able to trap greater than 90% of the CTCs to have a robust assay. You can see the sensitivity, if you have 5 million red blood cells per ml, and white blood cells are 3 to 8 million per ml, you only have 1 CTC per ml. So you need something to enrich for those CTCs in order to detect them. And unfortunately, in medulloblastoma, there isn't a universal surface receptor that we could naturally use to enrich for these cells. And there's lots of heterogeneity among the surface receptor expression throughout the groups. So this leads me to a day at Emory where Patti Seriolu from Georgia Tech in the Department of Electrical and Computer Engineering was giving a talk right here. And in his talk, he was describing a novel microfluidic chip to assess circulating tumor cells that used a label-free capture device. As you see here on the right, and I'll explain a little bit more, are trapped a cluster of circulating tumor cells. His focus is on clusters as the way to capture these cells physically. And clusters of cancer cells are important because they have increased viability in circulation and have been shown to have 100-fold greater potential for metastasis versus single CTCs. This is speculated to be because of their cell-cell adhesion properties, promoting survival and proliferation, their heterotopic nature that can occur. That means other cells, typically white blood cells, intermingled with the circulating tumor cell clusters. They have shear stress resistance and oikos resistance. They have properties of stemness and properties that allow them to escape the immune system. So Patti developed what the first generation called a cluster chip. Shown there on the extreme left was a chip that you would flow unprocessed blood from the patient through the device. And in the center panel shows an electron microscope image of the geometric pattern on the chip where cells, by the nature of their clustering, on the right shows 2 cancer cells together as a cluster, getting trapped within the device. And the normal blood would flow through the chip at a specific flow rate to allow this to occur. And he applied this to breast, melanoma, and prostate patients and saw that in these patients, typical clusters ranged from 2 cells to 20 cells per cluster, and up to 40% of the adult cancer patients had the presence of circulating tumor cell clusters. His subsequent device is from the cluster chip is now called Cluster Wells. And this is an improvement on the prior device because instead of flowing sort of horizontally, if you will, pushing the blood through the chip, this now can allow, by gravity, a syringe to gently drip the blood on to the chip that acts as a microfluidic and filter device, if you will. You see the chip here, and I'm not going to explain it in great detail because I'm not the engineer, but the chip was 3D printed in the clean room at Georgia Tech, and then subsequently could be made these plastic molds. So you can really make hundreds of these chips very cheaply and very quickly. And you see what happens is you've got essentially a punch through into the filter and the little holes that allow normal cells, single cells, to pass through, but clusters of cells get trapped. So after hearing Fadi's presentation here at Emory, I approached him and said, hey, we've got this GFP mouse and we've found these cells in the blood, and I'm interested in medulloblastoma. Would we be able to apply your chip to medulloblastoma? So we took the smoothen GFP mouse, 3 of them, and obtained the blood and then flowed the blood onto the cluster wells. And we found 3 out of 3 of the transgenic mice had the presence of circulating cancer cells easily detected through the cluster wells shown there on the right panel. So then the question came, can we do this in medulloblastoma patient samples, both blood and CSF? So first, the assay had to be optimized for the capture of a minimum 2-cell clusters so that we could, as the liquid biopsy rule goes, obtain greater than 90% of all the circulating cells that were present at any given sampling time point. So multiple different cancer cell types, including medulloblastoma, but not shown here, were artificially clustered and then with a known amount spiked into blood and CSF-like samples and then flowed onto the cluster wells. And in the figure shown in the bottom left, essentially the flow rate would determine how sensitive we were for detecting all of the cells that were spiked into the samples. And we want more than 90%, so we see that the top blue line, which is 25 mils per hour, is superior to the 100 or the 250 mil per hour flow rate for detecting at least 2-cell clusters in greater than 90% of the samples. PODI has further optimized this that I won't go into detail, but essentially we can use a faster rate and capture 100%, at least in the laboratory. Okay, so this then led to investigating in medulloblastoma patients through a V Foundation award in 2019 to see if circulating tumor cell clusters could act as a biomarker for medulloblastoma treatment. We performed serial analysis of blood. It was 10 ml of blood collected and 3 ml of CSF from 15 newly diagnosed and 10 relapsed medulloblastoma patients. We collected the blood at diagnosis. Now, it was post-surgery, but pre-treatment. We have since now collected prior to surgery as well, or at the time of relapse prior to any treatment, and then follow these patients every 3 months for up to 12 sample collections, or about 3 years. CSF was collected as clinically indicated, and we had 20 quote-unquote negative controls. I say quote-unquote, 10 low-grade glioma patients and 10 non-tumor subjects, but we only collected at one time point. We did find that no circulating tumor cell clusters were present in any of these 20 negative controls. However, we are suspicious that if we followed the low-grade glioma patients maybe further time points, that we may actually detect cells. Mind you, all the samples were blinded, so we would take the blood, we would give it to Georgia Tech. They wouldn't know if this was a control, if this was a medulloblastoma patient, and where they were in their treatment. Our aims were to determine the frequency and timing of circulating tumor cell detection in the blood and CSF. Second, to characterize these by cell number, composition, and molecularly by RNA sequencing. And third, to determine whether the amount, frequency, timing of the occurrence, or the morphological molecular character of these clusters correlates with clinical metastasis, response to therapy, and survival. So this is our patient cohort. We have a range of age from 7 months to 22 years, median 8 years. We have a roughly equal male to female population. All molecular subgroups were represented, although only one Wnt subgroup due to the rarity of that particular subgroup. And metastasis was present in almost half of all the patients, but 31% at diagnosis, which is representative of what we normally see historically for the cohort as a whole. And this is our flow diagram of the work, where we would collect blood and CSF, put onto the cluster wells, take the filter out, apply it onto a chip. The cells could be fixed and stained on the chip, and then automatic scanning to allow us to count the cells. So on the left middle panel are typical clusters that we would obtain from the blood. The staining for these cells, we replaced NCAM that Michael's group used with synaptophysin, because synaptophysin is not expressed by any hematopoietic cell lineage, but is restricted to central nervous system cells. Also, they needed to be negative for CD45 and, of course, nuclear DAPI stain. On the right is just an illustration of a particularly large clusters, and I'll talk about those in a minute. But those were rarely seen, but we saw those in a few patients. So in terms of how many we detected, on the bottom left are the number of clusters per ml. We would detect about 1 cluster for every 2 ml of blood. And the middle bottom panel shows the distribution of the number of cells within a cluster. So the typical distribution was about 2 to 5 cells per cluster. And they were, in the blood at least, the majority, 68%, were heterotypic, meaning they also contained a CD45 positive cell. And I believe you can see it here. I'm going up to B. You can see a CD45 positive white blood cell that is within the cluster of the cancer cells. The key findings from this is that we were able to detect clusters, which shocked me, in 100% of our patients, 25 out of 25, and we were able to do this in all patients with only 2 or more sample collections. I was expecting at the beginning that these would only be in the relapse patients and probably only those with either terminal disease or very aggressive, highly metastatic tumors. So in the blood, we detected overall 77%, or 10 out of 13 of newly diagnosed patients were positive for circulating tumor cell clusters in the very first blood sample post-surgery prior to treatment. And 80% of our patients we could detect overall. Of the total blood samples, 82%, or 127 out of 154 samples, had circulating tumor cell clusters. And this was on average about 6 ml of blood collected. Whereas only 25% of newly diagnosed patients had concurrent tumor visualized by MRI or CSF cytology. So in other words, there was no indication in these patients that there was any tumor remaining. In the CSF, we found 78% of patients were found to have circulating tumor cell clusters, and 96% of our patients, almost 100 there for 24 to 25, had circulating tumor cells with simultaneous blood and CSF single collection prior to therapy. So the importance of that is that if we're developing a biomarker using clusters, we need it obviously to be robust across molecular subtypes, and that we feel confident this could be used for all our patients. This would seem to indicate that we're able to detect clusters and then could follow them through the treatment and future follow-up of just about all our patients. We also found that cluster wells is 10 times more sensitive than CSF cytopathology for the detection of tumor cells. Of all the CSF samples, only 6.4% were positive for tumor cells by clinical cytopathology in the pathology lab. Whereas by the cluster wells, we actually detected 68% of the samples had circulating tumor cells within the fluid. So comparison of CSF to blood, we see that the clusters in the left panel look very similar to the clusters that we see in the blood. So morphologically, we didn't notice any difference. Also, the number of cells within the CTC, the distribution was quite similar, whether you were in the CSF or in the blood. However, we did notice there were less white blood cells associated with the circulating tumor cells. Only 21% of the clusters actually had a detectable white blood cell, which is probably reflective of the immune environment within the CSF being restricted. Going to panel E is a comparison of when we drew blood and CSF sample at the same exact point in time and looking for the positivity within each compartment. 60% of the samples had blood and CSF positive at the same time. 30%, the blood was positive, but the CSF was negative. 10%, the blood was negative, and the CSF was positive. And I said none of the samples where we collected together had negative CTCs in the blood or the CSF at the same time. And if you look at all the samples that we collected, 82% of the blood samples were positive, 68% of the CSF were positive. So it appears that at least between blood and CSF routes of circulation, they appear to be equally permissive. Now, whether they're contiguous or not is still a question. We were hoping to be able to discern that if there was a vast majority in the CSF or vast majority in the blood, then maybe we could speculate that the route is going from blood to CSF or CSF to blood. But this is still murky as to whether there's any particular primary initial route or whether it's all in circulation together. One distinct difference is, though, we looked at cell viability, looking at cleaved caspase 3, and the top is in the blood. You can see that the majority with the apoptosis, cleaved caspase 3 being yellow. You can see that the majority of these cells in the cluster, for this particular sample, not all of them, but it favored that the blood had more cells were dead in the cluster than alive. In the CSF, the opposite is true, that the majority of the cells in the cluster are viable. And this may make sense to everyone, since that's the microenvironment in which these cells live. And we know that leptomeningeal dissemination, not systemic dissemination outside the CNS, is what we see in medulloblastoma. So this is the favorable microenvironment for these cells and may be one of those steps, as I said, besides the genetic alterations influencing metastasis. Obviously, its interaction with the microenvironment likely plays a role as well. So what about CTCs and metastasis as defined by imaging? So we see that the positive patients with metastasis by CSF cytology alone was 12%, whereas for those demonstrating metastasis by imaging, 78% of patients had clusters of cancer cells in the CSF and 96% within the blood. What was interesting is the concentration of clusters per ml was clearly significantly higher levels associated with the metastatic patients, but only in the CSF. So again, reflective perhaps that it is the microenvironment that allows these cells to be viable and survive. On panel C, we looked at circulating tumor cell cluster size, whether you're 2-cell clusters or larger than that. And there was not a significant difference in size that observed in metastatic versus the non-metastatic patients. However, I'll show you a picture in a minute. We did observe, as I said, some very large clusters of 100 cells or greater, almost like microthrombi, which was startling for us to see and amazing that they were actually going through the circulation. The association of white blood cells within the clusters. We did see the higher association rate, so the more often that there were white cells associated with the clusters, the more you were likely to have metastasis. And this was seen both in the blood and the CSF. So now we have the three, at least major players who are thinking that regulating metastasis in medulloblastoma. You have your genetic background for facilitating. You have the levels of CSF increasing within the microenvironment that is conducive to their survival and proliferation, and then perhaps the role of the white blood cells further allowing metastasis to occur. So this is just an image of those rare, large circulating tumor cell clusters. There's no synaptophysin staining on this image, but just the nuclear in the CD45 negative for the white cells. And we saw these in both blood and CSF. And this is just a video I'll play in a second, but these large clusters were only observed in group 3 MYC-amplified metastatic tumors. So clearly there's something about this group of tumors, and these were seen both in the CSF and in the blood. And just to demonstrate, this is the cluster wells. The utility of it as well is that we're able to micro manipulate these large clusters off of the wells so that we can hopefully maintain their viability for further functional assays, definitely molecular characterization. But this is the hope that we can use this type of cluster wells to further define what these cells are and how they are acting mechanistically. So, as I said, we first wanted to molecularly characterize these cells. And in order to demonstrate, we again went back and spiked our wells with just cancer cell lines to prove that we could extract these rare clusters and then perform ultra-low input RNA sequencing on these cells, which we demonstrate preclinically. And in order to do this, you can't do this on two cell clusters, so you need to take these and group them together or pool the clusters in order to get a sufficient amount of RNA for the sequencing. So just pointing attention to Frank Chen, who was a neuro-oncology fellow in my lab and is now going to be assistant professor joining our faculty in neuro-oncology. And he was really interested in bioinformatics and applying his skill set to the high throughput sequencing data that we're generating from the circulating tumor cells from the medulloblastoma patients. And in the first iteration of this, we, again, didn't have a good control for it. And so we used our sequencing of our circulating tumor cells and compared it to a public database of normal hematopoietic cells. The point of that, we know that they're going to be different from her, but to demonstrate on the molecular level that they are clearly different from the white blood cells or any other circulating hematopoietic cells that we found in the blood. So the demonstration of that is the 40 significant top genes in panel D shows the distinction between our cluster cells and our white blood cells. And on the right, we show distinction again between these white blood cells and our circulating tumor cells. And it was interesting that at least on this comparison, CD34, a marker of stemness, was definitely increased in our clusters compared to white blood cells. And then reassuringly, we see markers. Oh, back up 1 second. I forgot to tell you that these clusters we wanted to define within a particular molecular subgroup. So if we put all the subgroups together and tried to define, it wouldn't make any sense. So we took our sonic hedgehog medulloblastoma patients. We took 3 patients, 4 different clusters of cells from each patient, pooled them, and then compared them to the white blood cells. So we saw that, at least in the public database, with the caveat of the batch effects that I'll address based on that. We saw reassuringly downregulation of genes we would expect to be downregulated in sonic hedgehog, MDM2, SUFU, patch, and TP53. So these were clearly sonic hedgehog cells that could be differentiated from the other normal hematopoietic cells. Now, to get around batch effects, because we know that in the public database, we're talking about huge numbers of cells, so the RNA levels that are being sequenced are enormously different. In order to address that batch effect problem, not all of them, but some of them, we then took PBMCs with equivalent cell numbers to the clusters and compared those in terms of sequencing. So at least we're working on the same RNA levels. And in the principal component analysis there on the right, we clearly see circled in red, our circulating tumor cell clusters are distinguished from the PBMCs on the RNA sequencing level. And then if you look at the top differentially expressed genes now, we get a very clear picture. A lot of these genes were retained even when we compared it to the public database, but the #1 overexpressed gene universally among all the sonic hedgehog medulloblastoma cluster cells is metastasis-associated lung adenocarcinoma transcript 1, or MALAT1, which is first discovered, as you might expect, in lung carcinoma metastasis, and is highly conserved, nucleus-restricted, long non-coding RNA that is upregulated in and highly associated with numerous cancers, including medulloblastoma, and is associated with poor prognosis in a number of cancers. And also, interestingly enough, has been described as a partner in GLI1 fusions and promoting activation of sonic hedgehog signaling, which would fit that these are sonic hedgehog circulating tumor cells that we've found. And the one thing I'll just briefly say, still caveat, we're comparing these to white blood cells, and our plan is to sequence the cells at the same time as we're sequencing the same number of tumor cells from the primary tumor to see if these cluster cells are distinguished from the primary tumor. Also, same number of cells from that patient, blood, and normal cerebellum if we can do that. So that's in the works. We're actually preparing that right now. And then we looked at markers of stemness, and clearly the sonic hedgehog circulating tumor cell clusters are overexpressing markers of stemness shown there, and are enriched for pathways that seem to promote extracellular matrix adhesion type interactions. And it goes back to our three components. So genetic background, markers of stemness, interaction with your microenvironment, and perhaps white blood cells. These seem to be the components that are necessary for your final clinical metastatic phenotype. And the question in my mind, are sonic hedgehog CTCs MALAT1 overexpressing cancer stem cells? This has yet to be addressed through our interrogation. Now, what about the white cells that were captured in the clusters? So Frank used CyberSort to do an immune deconvolution to try and ascertain what these white blood cells may be comprised of. And so we saw that gamma delta T cells and naive B cells seem to be associated more with our clusters when we compared the expression, when we compared the mixture to whole blood. And activated mast cells, macrophages, and dendritic cells. And it seemed to be shown on the left that macrophages were most clearly associated with the circulating tumor cell clusters. So we see that association. We still don't know what their function is and why they're there and how they get there, whether it's at the site of the tumor or whether it's in circulation that these cells become adherent to the clusters. I don't think it's an artifact. I think these are real and are doing something with these clusters. And then next we looked at the systemic immune cells outside of the clusters, but what's going on when the circulating tumor cells, and we enlisted talented postdoc in Rafi Ahmed's lab, James Ross, to perform multiplex flow cytometry of the blood at the same time as we were collecting the circulating tumor cells. And what he demonstrated is that as the levels of circulating tumor cells increase, they seem to directly correlate with peripheral PD-1 positive, CD28, CD8 positive T cells, which are the dysfunctional or considered, I'm not an immunologist, but are considered the dysfunctional exhausted CD8 cells that are incapable of mounting a sufficient anti-tumor immune response. So it appears perhaps this could be one way of tumor immune evasion is through circulating tumor cell clusters. Again, this has to be completely interrogated further to confirm it. All right, so going to end with these last slides, which is really the title of my talk. So after all that, we get to this point where I don't expect anyone to look at these and decipher anything. It's just to show you the amount of time and effort we collected over 3 years, serial collection of blood and CSF in all 25 of the medulloblastoma patients. And what was interesting is that I would stare at these graphs for hours as we went along, and they really didn't make any sense to me at all. And I thought, oh, well, it's great we can detect them, but I don't think there's any utility to these cells to give me information of what's going on in the patient and how they're responding to their treatment. But it took 3 years to actually get enough data to show survival confirmed, relapse confirmed, to see a pattern that emerged from all of these data. And it's illustrated here. So I'll try to focus on this one slide. It's really the take-home message, is that we do find that the concentrations of circulating tumor cells appear to have a biomarker significance in terms of response. So what this shows you, we'll start on the left, is in red are the CSF, circulating tumor cell clusters that we've detected, and in black are those in the blood. And the colored shaded areas are the types of treatment they're receiving, the patient. So yellow is chemotherapy, pink is high-dose chemotherapy. We typically use that in infants to spare the use of craniospinal radiation. And over here is green, which is the radiation therapy. And the first thing that we notice is this patient is positive for clusters at diagnosis, but immediately upon induction chemotherapy goes to zero and stays at zero, undetectable now through our assay. Conversely, and this is what really bothered me for some time, is we would see a spike in the number of the concentration of circulating tumor cell clusters in the blood following treatment. And it was usually following not standard conventional therapy. You see that there's not really change here over baseline at diagnosis, but they hit the high-dose chemotherapy, which is what we use in place of radiation therapy. And that's when we see this spike. And eventually, the cells continue to circulate around. And this is 40 months since diagnosis. This is the longest we saw. But they eventually, when you see this type of pattern, they eventually trickle down to undetectable and stay undetectable. And this is an example of a long-term survivor. So we feel that this spike over baseline is a marker of treatment response. Now, what's going on? I don't know, whether it's breaking down the blood-brain barrier and these cells are passively being extruded in clusters, or there's a mechanism for this. But right now, we see this pattern as a marker of response. Similarly, you need to have these cells go down immediately in the CSF, and you need to have them go to zero in order to be declared a good response. Now, this is a patient who relapsed, and you see the opposite occur. At baseline in the CSF, we detected cells, and during treatment, following treatment, there's a significant increase in the concentration in the CSF. And this never, despite additional treatment, never goes to zero. Likewise, in the blood, we see that actually the concentration goes down and never has that spike we see here and stays down, only until we get to a palliative salvage radiation regimen do we see a bit of a spike, but it then goes down again and the patient relapses. So we think this is a marker. If the cells in the CSF go up and there's no marker or indication that the cells are going up in the blood, that this is a treatment resistance and portends possibly that this patient will relapse down the road. And mind you, MRI and CSF cytology throughout this treatment is negative prior to relapse. So the only detection of tumor still remaining was through our circulating tumor cell clusters. And so of the 12 newly diagnosed patients, all 10 long-term survivors demonstrate the pattern on the left, while the 2 who relapsed demonstrated the pattern on the right. All 9 relapse patients demonstrated the pattern on the right during their treatment, and all of them subsequently progressed and died, with the exception of 1 patient who showed a pattern on the left after relapsing in treatment, and is the only survivor among the relapse patients. So that's our preliminary data that we hope to put into a new grant application to validate this, which is obviously the most important step since the sample size is still quite low. But the conclusions are that circulating tumor cell clusters are novel and robust biomarker for minimal residual disease in medulloblastoma, and is the only tool we have currently that exists for this purpose in this disease. and that serial changes in circulating tumor cell levels appear to correlate with tumor response, treatment resistance, and relapse. Higher levels of clusters in the CSF and the presence of white cells within the clusters appears to correlate with metastasis. Very large clusters were only seen in group 3 MYC amplified metastatic tumors. So again, going back to that MYC amplification, there's something about it that influences this particularly large group of clusters. It wasn't seen in all MYC amplified tumors, but it was only seen in those patients who had those tumors. Circulating tumor cell clusters, we believe, can be used to track tumor evolution with treatment. Another feature that's perhaps different from looking at circulating tumor DNA for evolution, because we're able to capture the entire genome within these cells rather than fragments of DNA. And of course, in the blood, circulating tumor DNA for brain tumor patients has been fraught with lack of sensitivity. And the cluster wells chip technology enables us to capture living cancer cells from the blood and CSF so that we could do additional functional assays on these cells. The cluster wells assay could potentially be, in my mind, paradigm shifting for disease monitoring in these patients and helping to guide therapeutic decisions. We are also applying this on now multiple. We have another grant to look at other brain tumor types besides medulloblastoma. So we've covered the entire spectrum of childhood tumors. Would love if we have more grant funding to extend it to the adult Winship population to see if in glioblastoma we could use this same strategy. And our future directions, as I said, number one is validation. I'm going to propose this to the Pediatric Brain Tumor Consortium, which is 12 centers across the US, to see if they'd be willing as a biologic protocol to send us blood of patients undergoing investigational treatments where we could track circulating tumor cell clusters to have an indication whether they really do predict relapse, response to treatment, and treatment resistance. The second major is taking these cells, trying to propagate them in vitro, doing drug testing, which we've done on one, and we have seen that the majority of drug responsiveness between the primary tumor cells and the circulating tumor cells, they're very close, but there are some drugs that are completely resistant only in the circulating tumor cells. So again, is that the possibility for why we see these patients relapsing 10 years later, is that these circulating tumor cells are the maybe cancer stem-like cells that are quiescent for long periods of time, drug resistant, radio resistant. And if we could find out if they are the hallmark for relapse, then perhaps therapeutically targeting the circulating tumor cells will allow us to get better survival outcomes. Okay, and with that, I will close a little early and just thank all the people who were involved, and highlighting the first names that are listed. I said from my lab, Frank Chen, from Georgia Tech, Tevita Oskaya, and from Emory Winship in Rafi Ahmed's lab, James Ross, and I just also list Manoj Bhasin, who has helped us as well with Frank in terms of the bioinformatics of the circulating tumor cells. And I would be remiss if I didn't thank Rachel Gray and Pushali Bhattacharya, who Rachel's a physician assistant, and Pushali is the clinical research coordinator who assisted us with the collection of these samples over 3 to 4 years. And the picture there is the tip of the cap to Scottish Rite, since we are now no longer headquartered there. We're in our fabulous new Arthur M. Blank Hospital, but that's where the magic happened for this entire circulating tumor cell cluster study. So thank you very much for your attention. Oh, okay. Yeah. Okay. See if I can read these. See, do you think you could find these clusters in lymphatic fluid? Sorry, I missed it, but what is the volume of CSF and blood needed for your test? So the latter, we collect 10 ml of blood and typically 3 ml of CSF, which is sort of standard clinical practice. Lymphatic fluid, I would see why not. I'm not familiar with extracting lymphatic fluid from patients, so I would need to be instructed, educated on how that might take place. But yeah, I think any place in which these cells could possibly circulate, you could at least investigate if they're there. Second question, were you able to see CTCs in blood in non-metastatic settings? Yeah, so absolutely, that 100% of the patients, and that was the shock to me, is whether they had metastasis or not, all medulloblastoma patients appear as part of their disease to actually have circulating tumor cells. And we saw these, again, after just the first 2 sample collections. So you were diagnosed, we either found it on your first sample after surgery, or we found it on your second sample 3 months later in all of the patients. So circulation is not metastasis. And what's interesting is if we find cells by CSF cytopathology clinically, we deem the metastatic patients and we automatically bump them up to the higher risk, high dose craniospinal radiotherapy. And based on these results, I would argue that may not be the best thing to do because that's what these cells are doing naturally, but that's not metastasis. Other steps need to take place in order for clinical metastasis to occur. And any role for a non-invasive diagnostic tool. Not sure exactly what you're referring to, non-invasive diagnostic tool. We would think this is minimally non-invasive, blood, CSF. For the CSF, we've considered a lot of our patients actually get Omeyas placed as more investigational therapies are now, particularly immunotherapy, are looking towards delivery of therapeutics. So my thought is that we could use this already implanted device to serially collect. Again, it'll be a needle into the Omeya, but that's minimally invasive. Sorry, I'm not sure what non-invasive might be. Yeah. No.
Thank you. You may have mentioned this, sorry. Yeah. In the adult side, specifically in lung cancer, and this is circulating tumor DNA, is use it to adapt therapy. In other words, you start with baseline detectable levels, you treat the patient. Within a short time, you look at whether they clear or not. And if you have persistence to the even if they have a clinical response, they're not going to have a robust clinical response. Maybe that's an early point for intervening before effective therapy.
Yeah.
With circulating tumor cells, do you see some sort of an applicability? Are there any data on looking at clearance of therapy and using that as a biomarker to say it's from the
Yeah, no, definitely. That's this slide that trying to illustrate that, at least in the CSF for sure, this is the red dot. It appears if you don't clear these cells, this is a diagnosis immediately with some sort of therapy, that these are the patients prone, if you don't clear it, prone to relapse. This is a good response. This is an illustration of what happens during therapy. We see an escalation actually in the CSF of these cluster cells, and all the patients who demonstrate this have relapsed for us. And all those who have had good responses, and this is quick, within a week, we can actually see the CSF cells go away. So, so yes, and also secondary, as I said, since we're getting the whole cells out, we can also look at the whole genome theoretically. It is challenging because of the low number of cells, but we are attempting to do that now so that we could also partner just with cell numbers with the DNA evolution. So we think also with treatment that we may be able to use these cells to watch how expression is changing or the DNA, if new mutations are arising by tracking these cells during treatment. And I didn't mention it, but in our grant, we also put that with the advent of immunotherapy, particularly cellular immunotherapy, and I showed you white blood cells are engaged with these clusters, whether we could actually use that as a monitoring tool to see if the cellular therapeutics are engaging as they're supposed to with these cancer clusters over time. So I think we have at least 3 different modalities we could use to track monitoring of response. No more questions there.
I had one question.
Oh, yeah.
curious about, if there are tumor cells in the blood, they must have migrated through the blood-brain barrier.
Yeah.
About how neuronal cells do that with lymphocytes. Corollary to that, do you think lymphocytes are necessary to be seen in the cluster? Do they somehow facilitate them in or out of the CSF?
Yeah, maybe that's a great point. That could be one of the ways they're kind of transporting these clusters out. As I said, because we see the association of these cells with metastasis, I do think they're doing something, and that's the next, preclinical evaluations is to look at this in the mouse and to sequentially whether we knock out the white cells and see what happens or we alter, And I think it's again alterations of the tumor cell itself with the microenvironmental cells. Those would be the key changes.